Well, hi, everybody. Good to have you with us. We're ready to begin Chapter 2, entitled Tools of the Laboratory. This is really perfect timing because we're just beginning to learn some of these different laboratory instruments and how to use them coming up soon. So this is just a good time to get into Chapter 3. So let me begin as I always do, sharing the PowerPoint. This, of course, is found in the course shell. Start with the cartoon. These Gary Larson cartoons are great. He has discontinued doing these as of about, I don't know, 15, 20 years ago, but a lot of them had biological connotations to them, and you have to be a bit of a nerd, I guess, to appreciate them to the full extent. You're going to see another one coming up soon, too. But I think they're kind of fun. Okay, this chapter begins by kind of providing an overview of what are referred to as the 6 I's of microbiology. Let's just kind of go over those briefly, and we will be spending more time in this chapter talking about them in more detail. But a lot of these sort of make sense from a chronological point of view. You know, the first thing that we do in the laboratory is we inoculate. We introduce bacteria onto or into different types of media. Here we see an individual with what's called a loop. This is a disposable plastic loop. You're going to be using some metal loops that are going to be flamed and can be reused. But this particular one, once he or she is done doing this streak plate, this would be disposed of. They would obviously not be able to reheat that and reuse it. So inoculation, again, introducing a bacterial sample onto the surface of a plate of what's called agar. You'll be seeing that coming up very soon in lab. Sometimes we use tubes, basically test tubes with caps that contain a liquid material. We call it a broth. So you're going to be seeing these coming up actually this week in lab, if you haven't already seen it. We will inoculate the sample into a medium that will support its growth. Now, in order to kind of promote the growth, we typically then place the inoculated media into an incubator, where it is allowed to sit for a day or two, depending upon kind of what we want to grow and what the bacterium is that we're trying to culture. We have several incubators in the laboratory. And the ones that we'll be using 90% of the time are incubated at 37 degrees Celsius, which I hope you know is body temperature. We will be doing a lab toward the end of the semester where we place bacteria into different temperature regimes. We use, I think, 3 or 4 different incubators at different temperatures, and we see what effect temperature has on growth. So we'll be talking more about that in an upcoming chapter. Oftentimes, we want to isolate bacteria, and we can do so if we perform what's called a streak plate. You'll be doing that in lab in a couple weeks as well, and you heard me allude to that in one of the preceding slides with the plastic inoculating loop. Remember, that person was streaking the plate. And once that streak plate is pulled out of the incubator, this is typically what you like to see. The idea is to spread out the bacteria in ever decreasing numbers, and we'll talk about the methodology and technique used to do that. The goal here is to get isolated colonies, and so these circular white structures you see here are individual colonies of bacteria that arose from a single cell that was eventually, spread out on that plate, and as that cell underwent binary fission and division, a few days later, it grew to a very sizable number to the point at which we can actually see it with a naked eye as this little circle. Now, this is indicating that we can then subculture this and grow it in pure culture. Remember, we said earlier that pure culture was defined as a culture that had just a single species of microorganism, we'll say bacteria, growing in that broth or on that slant or whatever the medium might be. That was an important one of Koch's postulates, if you recall from chapter 1. This is just a sort of an artistic version of plating, and you can see some of the isolated colonies here. I just threw that in for fun, but you'll be doing a streak plate, as I said, in a couple of weeks. So we often want to isolate the bacteria because when you get a sample from a patient, it's a mixed culture, isn't it? And so we need to separate those bacteria out to the point at which we have isolated colonies, and then we can do more study on those to determine exactly what the identity is, say, of that mixed culture. But we have to get isolation first. We can then inspect those cultures once they are pulled out of the incubator. We will be assessing them in two different major ways, at least visually. One here is using our naked eye to look at the colonies. We're looking at their color, we're looking at their shape. There's a whole series of different characteristics that we can assess and quantify simply by looking at those with our naked eye, or maybe using a dissecting scope, which we'll talk more about in lab. I'll show you that coming up soon. And you can see from this plate that we've got a variety of different colors and different sort of textures, if you will, to the margins of these colonies. These actually look somewhat like fungi. Kind of hard to tell exactly what they are. Could be bacteria too. We're going to also spend a lot of time in lab looking at these under the microscope, and we'll be talking about microscopy in just a few moments here in chapter 3. We'll be staining our cells to allow us to differentiate between different types of cells. For example, gram-positive and gram-negative cells are examined under the microscope, and you look for specific colors to appear. Or you may be looking at particular structures that the bacteria might have, say flagella, or the presence of, say, an outer capsule. So microscopic analysis is an important integral part of microbiology. And as I said, you're going to be spending lots of time with the microscope in lab. So we want to be very proficient and comfortable using it. I mentioned earlier that one other project that you'll be doing in lab this semester is doing a report of the unknown, right? I'll be giving you an unknown bacteria. You'll be looking at it under the microscope. You'll be staining it. You'll be subjecting it to a bunch of different tests. We'll inoculate that unknown into a wide variety of media, some broths, some semi-solid agars, and so forth. And the idea is then to come up with an identity of that bacteria. So we're going to be gathering lots of information. You will be doing that. Then hopefully identifying that unknown. And of course, that's an integral part of any diagnostic process at the hospital. And a patient comes in sick, they want to be able to isolate the bacteria. They want to be able to identify what is the problem pathogen. And that, of course, will dictate what sort of antibiotic perhaps might be prescribed, or what mode of therapy might be then instituted within the hospital setting, or maybe a medicine prescribed to take home and take it, whatever the case may be. So we will be using a variety of identification techniques, which we'll again talk more about coming up in about a month and a half or so. The proper identification is a critical part of these eyes of microbiology. In fact, it's the last one. So here's just a review of those 6 eyes, starting with inoculation, incubation, isolation, inspection, gathering information, and hopefully culminating ultimately in the identification of the bacteria. Just a nice summary slide to take a look at, spend some time reading. Well, as I alluded to a moment ago, This instrument, the microscope, is one that we will be spending a lot of time using. And you've spent 2 semesters at least with it in ANP1 and ANP2, so I know you're somewhat familiar with the microscope and what it does and sort of how it works. The goal is to, of course, magnify and see structures, but also to have good resolution. The ability to see detail is critical when looking at bacteria, for example. Here's another Gary Larson cartoon. Yes, microscopes were big back in the Stone Age. See those woolly mammoths? We'll be using the compound light microscope in our lab. This isn't exactly the same scope that we have, but most compound light microscopes share similar structures and parts. Obviously, here you see the 2 ocular lenses. We have binocular compound light microscopes as well. Here is the nose piece that houses our 4 objectives. You can't quite see the scanning objective. It's hidden here behind the oil immersion objective. And then the stage that we'll be placing our slide onto, and a little bracket that will hold the slide within this metal part of the stage. We have a light source from below that we can regulate the intensity of using a little dial here on the side. There's also usually an iris diaphragm within the condenser that can regulate how much light is allowed up through, in addition to the intensity of the bulb. And then, of course, we have the ability to both focus with coarse and fine adjustment. This is going to end up raising and lowering the stage. So we'll, again, review that coming up in the lab in a few days, if we haven't already done that. You might also remember that it's easy to calculate the total magnification of the object being viewed under the microscope if you know 2 things. One, the objective magnification, as well as the ocular lens magnification or power. Ocular lens magnification is always 10x. What changes is the objective being used, right? So again, easy way to calculate total magnification. Take 10 times whatever objective you're using. That'll give you the total magnification. Resolution is another important aspect of microscopy, this ability to see detail. And the book describes this by asking you to envision the utilization of longer wavelengths of light, as evidenced by the larger yellow circles here that outline the periphery of one's hand. So large wavelengths of light are not good to use if you want to see detail. And so this view of the hand does not illustrate the fact you have 5 fingers very well, does it? It does indicate a general outline, but that's about it. If we were to utilize smaller wavelengths of light, as evidenced by the smaller circles now, we would be able to pick out the 5 fingers. We would obviously identify that as a hand. And that's exactly what the compound light microscope and other microscopes do. They utilize small wavelengths, short wavelengths of light. In fact, in our microscopes in the laboratory, there is a blue filter that's housed in the condenser. And that's there to allow us to utilize the smallest wavelengths of visible light that we can. And that happens to be the blue and violet end of the visible light spectrum. You might remember a rainbow is made up of a variety of colors, ranging from red-orange on one end of the spectrum to blue-violet on the other end. The orange-red are the larger, longer wavelengths of light. And then as you proceed across that visual continuum to the blue-violet end, then we come to the shortest wavelengths of light. So utilizing that blue filter in the microscope is basically allowing us to use, again, those short wavelengths to provide us with the maximum ability to see detail. When I say detail, I'm not talking about your hand. I'm talking about seeing detail as it pertains to perhaps an outer capsule of a cell or other perhaps internal structures of certain cells if we employ specific types of stains. And the way that we can do that in the microbiology lab is by the utilization of the oil immersion objective. And so in this sort of insert photograph that I stole from the internet, here we're zooming in on that area where we've got our oil immersion objective. And here's our slide, of course, on our stage. As we zoom in then to the center of the slide, here is the oil immersion objective. Here's our slide. And this is kind of comparing and contrasting what happens to the light that comes from the lower light source. Okay, the bottom of the microscope, you've got that lamp, right, that you can regulate the intensity with by turning that dial. If we don't utilize oil, and I'll walk you through the steps of how to properly do that in lab coming up. But without the utilization of oil, you'll notice that while some light rays that pass through the clear slide from the condenser do make their way up into the objective and into your eye, as evidenced by these two yellow arrows, right? There are a considerable number of light rays that would diffract off the glass slide and be lost off to the side here of the microscope and not go up into the objective. They'd be sliding off this direction, that direction. And there'd be other light rays that would diffract off the top surface of the glass slide and bounce downward away from the objective, okay, off to the side. So there's a considerable percentage or proportion of light rays that are not going to be bent in the proper way to go up and enter into your eye if you don't use oil. Okay, so when you use the scanning objective, the low power and the high power, those three dry objectives, if you will, you do lose a considerable number of light rays, and therefore the resolution is not super great. It's not bad, but it's not as good as it could be if we used oil. So on the right-hand side of this slide, where we have now employed oil, theoretically, between the objective and the slide, notice what happens to those light rays that would ordinarily be refracted and diffracted off to the side and not up into the objective. Well, they're bent, aren't they? So the oil has a particular property that bends the light rays so that a greater percentage of them go up into the objective and up into your eye. That results in much better ability to see detail, or to have a finer resolution, basically. So the use of oil immersion allows us to see structures that are 1,000 times actual size. Okay, how do I know that? Well, I know that the ocular lens, remember, is 10x. So let's go back to that preceding slide. We don't see the ocular lens up here, but you know that's the thing you're looking through here. This is 10x. And if we're utilizing the oil immersion objective, we're utilizing an objective that has a magnification of 100. So 10 times 100 is 1,000. So that is the limit of what we can see with our compound light microscope. So basically, this slide is showing that this region here in kind of purple, where they say resolvable, this is what you can see with our microscope. Anything in the blue area, which is smaller than our ability to see these things because of the size, we can't see viruses. We cannot see certain types of prokaryotic cells called mycoplasmas. These are viruses up here too. We can't see bacterial flagella. We can see the cell, but the flagellum, or in this case, two flagella, we just don't quite have the resolution to see those. So I'm going to have you think back to chapter 1 when we talked about units of length, and we described, millimeters And we said that there were 1,000 micrometers in a millimeter. Remember that? And there were 1,000 nanometers in a micrometer. Well, this is depicting micrometers, this kind of funny U-shaped symbol. So there's 1,000 of those in a millimeter, and you can look at any ruler in your drawer. That small unit that you see on a ruler or a meter stick, that's a millimeter. So if you divide it up 1,000 times, you would have a micrometer. So we can get down to 1 micrometer or maybe even less than a micrometer in size with our compound light microscope using the oil immersion objective. But anything less than a micrometer or half a micrometer or a tenth of a micrometer would be beyond really our ability to see that, to resolve that with our scopes. So we utilize what's referred to as bright field microscopy. Again, this is basically an artificial light source that beams the light up through the slide, up through the specimen, up into the objective, and ultimately into your eye. And this is generally fine for looking at most, specimens, whether they're bacteria, although again, we have to use oil immersion for that. But remember, in A&P, we looked at slides, prepared slides of different kinds of tissues, right? We had no trouble seeing muscle cells or skin cells, and we could see the nuclei inside those cells and so on and so forth. So this is the most common type of microscopy that you would find in high schools and colleges. But if you go to a university, you might have access to an electron microscope. These are very expensive instruments. They used to be really, really large. I mean, some in the early 30s, when these were first built in Germany, were the size of refrigerators. They were very, very large. Today's electron microscopes are probably no larger than a small microwave in terms of overall size. So they've come down in size, but they're very, very expensive. They'd be used in research for the most part. But what electron microscopes make use of, as dictated or inferred by the first word here, is they don't use visible light like the bright field microscopy that we talked about does. They make use of these subatomic particles called electrons. And basically, when you look at the wavelengths of electrons, we're talking about something that is, as it mentions here, 100,000 times shorter than the shortest wavelength of visible light, which remember is the blue-purple end of the spectrum. So this greatly enhances our ability to see things that we could never, ever see with the compound light microscope using bright field microscopy techniques. In fact, electron microscopes allow us to see things upwards of 1 million or maybe even more than 1 million times actual size. It's mind-boggling. There are some electron microscopes that can see 5 million times actual size. So it's phenomenal what these instruments can do. And I want to just briefly talk about 2 different types of electron microscopes. The first of which is referred to as the transmission electron microscope, which has the ability to take what's called a TEM, which stands for a transmission electron micrograph. So it's basically a photograph of what the scope sees. And what a TEM will allow us to do is to look into a cell and see internal structures within cells. Now, these could be bacterial cells, or you can take TEMs of eukaryotic cells too and examine, let's say, the rough or smooth endoplasmic reticulum within a cell. Or you could look at the flagellum of a sperm cell. All right, so it's not exclusive to prokaryotic cells. You can look at anything you want with electron microscopes. So here's what a TEM would look like of COVID-19. Now, I probably should have made more careful note of what the magnification was of this particular TEM of the coronavirus. But what it does allow us to see are those glycoprotein spikes here on the surface of the virus. You've seen diagrams on the news and in social media, it has these kind of orange or red or blue colored spikes. They just color code these so they're easy to see. They're not really blue, but these are the glycoprotein spikes. This is how the virus basically then docks with the host cell. We'll talk more about viral biology coming up in another chapter, but you can see some of the outer structures here. Not super great because this is a very tiny entity. The other type of microscope, electron microscope I want to mention, is the scanning electron microscope, which takes SEMs, scanning electron micrographs. In some respects, these are the more fun photos to look at, I guess, and I'll show you why in just a moment. But in essence, what you're seeing here is not internal structures so much as it is external structures of whatever it is you're looking at. And that's because the technique and the technology behind these is different than a TEM would be. So here we're looking at a scan electron micrograph of some bacillus-shaped, rod-shaped bacteria whose genus is Schuanella. That's how you pronounce it. I'm not familiar with Schuanella, but there's what it looks like, and you can see that these definitely give you a sense of almost, I won't say three-dimensionality, but sort of like three-dimensional. You can see that there's depth and length and width to these cells. We use special stains and dyes to color them. Schuanella isn't really purple-blue, but it definitely is a bacillus-shaped bacterium. If we click on this link, and let me just go back and just make sure that you can see these. This particular website has some really fun SEMs of different structures that I thought you might get a kick out of looking at. Here's pollen from different kinds of flowers, including sunflower, primrose, and lilies. In A&P, we talk about the pseudostratified ciliated columnar cells that line our trachea. Well, here are the cilia, here emanating from those epithelial cells. Again, giving you a three-dimensional surface view. There's a head and mouth of a leaf beetle. The eye of a fruit fly. Insect eyes have compound lenses. So each one of these little circular structures is an individual lens. You have just one lens in each of your eyes. Insects and other arthropods have what are called compound eyes. They have many thousands and thousands of lenses. And that's why it's hard to catch a fly. You move your hand, they detect that movement because they have lots of lenses to pick up on movement around them. Here's a mite. There's a bed bug sticking its tongue out at you. Again, a lot of these have been colored just to bring out particular features. Surface of a kidney stone. Very crystalline, right? Yeah. Anyway, I'm not going to go through all these. Check it out. It's a really fun website that depicts, again, lots of different cool SEMs. Okay. In lab coming up, we're going to also be performing stains on some algae. And we're going to be utilizing what's called a wet mount, making our own wet mount. So this is a common technique that one can use to look at leaves under the microscope. Take a leaf off a plant and add it to your slide and examine it. And it's a very simple process. All you do is you take a clean glass slide, put your specimen, maybe a leaf, if it's a structure you want to examine. It could be insect, butterfly wing, maybe you put down there, or it could simply be a drop of pond water too. But you've got to add some sort of liquid suspension to your specimen. If it's pond water, it's already liquid. If it's a little piece of leaf off a tree, you'd stick that on the slide and you'd put a drop of distilled water on the leaf. And then you would add a cover slip on top of that. And that basically would result in the formation of what's called a wet mount. It's very, very simple to do. And then you would examine that under the microscope. Another technique that we really won't be doing, but it's sort of fun to talk about, is what's called a hanging drop mount. And this utilizes what's called a depression slide. So in these depression slides, as the name implies, there's a little kind of cut or depression that's been made in the glass slide. And what you basically do is you take a cover slip, you put a drop of suspension, let's say it's pond water. You put the drop on your cover slip. That's that little plastic square that you've perhaps seen in the bio lab. Okay, so just kind of picture what I'm describing. A drop on top of the cover slip. You take some Vaseline and using a toothpick, you apply that around the periphery of your cover slip. Usually you go at the corners. And then you take this depression slide and you turn it upside down. Okay, so the depression is kind of in an arch sort of format. And you would lower that down onto the cover slip that has your drop of pond water, so that the drop is in the center of the depression. You just gently lower it down, let the Vaseline make an adhesive connection with the cover slip in the slide, and then you take that slide and you invert it, you flip it, and that's what you're going to see here on the diagram. And the purpose of using wet mounts, you might say, what's the difference between wet mounts and hanging drops? Well, the hanging drop slide has the advantage of allowing you to see more natural movement of organisms, let's say protozoa, maybe single-celled pond critters, in that drop of water, because there's no pressure being applied to them. With the wet mount, remember, you have the cover slip pushed down onto the slide, so the organisms are sort of getting squished a little bit. Here, they're not. And so the idea here is that you can kind of observe them in a more natural way, that they're able to swim around and so forth. So just kind of a fun technique. The times that I've used it mostly in the lab is looking at pond water. We're going to be making some slides using bacteria coming up very soon. And typically what we do is, if the bacteria is coming from a liquid broth, we would apply that to the slide and let it air dry. If the sample was coming from a semi-solid material, like a plate, remember we looked at the plates earlier in this chapter, PowerPoint. If you take the bacterium from a solid or semi-solid material, you would have to first add sterile water to your slide, and then add your inoculum, your loop full of bacteria, for example, and you'd mix the bacteria with the water, because if you didn't do that, there'd be just too many cells. It'd be way too concentrated. So the idea is add a couple loopfuls of sterile water first to the slide, then add a loopful of bacteria, and then mix that a bit, spread it out on the surface of your slide. That's allowed then to air dry. And usually, once you've allowed the slide to air dry, in most staining procedures, you would then heat fix the slide, it's called. So heat fixation involves taking an air-dried smear that you've just performed or made on your slide, and you pass that slide through a gentle flame of a Bunsen burner. And as the slide mentions here, this heat fixation does two things. It kills any cells that might still be present, and it also helps the cells stick to the glass surface of the slide. And you might wonder, well, why would we want want the cells to stick to the slide. Well, remember, we often want to stain those cells so that we can see them, right? We can see different structures, let's say, of the cell. And so heat fixation allows us to have those cells stick to the surface of the slide, because if we don't somehow help them adhere to the surface, when we add the liquid stain, they would get washed off. So that's why we heat fix. Now, as it mentions here, we want to be careful, though, that we don't allow that heat fixation to be too intense. The flame has to be a sort of a gentle flame, and I'll show you how to adjust the Bunsen burner to do that. So you don't want to heat fix too much, otherwise you're going to have charred cells that will be very distorted. You won't see anything. But you want to heat fix enough so that they stick to the slide. So it just takes practice, and you'll get plenty of practice with that coming up soon. So Robert Koch, our German bacteriologist that we talked about in chapter 1, really is noted for his staining techniques that he established back in the 1800s, and we use many of those same techniques today. So we're going to be using all different kinds of stains or dyes. Different dyes are used in different ways to bring out different features within the bacteria that we want to take a look at. And the first type of stain I want to talk about is called the positive stain. And the easy thing to remember about positive stains is that these stains will color the cells, and the background will be white or clear. So if you look at this photograph, we're seeing the purple coccus shaped spherical cells here and the white background. Let's talk a little bit about how a positive stain works. Let's talk about the actual chemistry of the dye that's used to make a positive stain. And we will be employing what's called the basic dyes. There's acidic dyes and there's basic dyes. Here we're talking about basic dyes. And we're going to be describing a particular part of the dye molecule called the chromophore of the stain molecule, the chromophore. And in the case of basic dyes, their chromophores, as I've indicated here, are positively charged. They're cationic, positively charged chromophores. So if we look in the lower right-hand corner of the slide, here we see the outer surface of the cell envelope, it's called, in brown. And we have to understand that in the case of most bacteria, the surface of the cell envelope here contains negative charges, negatively charged outer surfaces here of the bacterial cell wall. Here's our positively charged chromophores connected to the stain molecule. So just think of these positive charges as the dye, the stain. These are basic dyes, basic stains with positive chromophores. Now, what do you know about unlike charges? Well, they attract, don't they? And so here we see the positively charged chromophore of this basic dye, illustrated by the blue circular plus charges, connecting to the negatively charged surface of the cell wall of the bacterium. And that's why the cell itself is colored, giving us a clear background. Okay, so that's the overall effect. Positive stains employing basic dyes, color the cell, background clear. Contrast that with an acidic dye, okay, whose chromophore is negatively charged, also referred to as anionic. So you already have the negatively charged outer cell envelope, cell wall here of the bacteria, and here you've got the negative charges associated with the chromophores of the acidic dyes. These are not going to complex together, are they? Unlike charges, repel. So in this particular instance, the cells will not be colored, the background will be colored, as illustrated by this photograph, right? So here we see the bacterial cells in white. They appear to be rod-shaped bacillus cells, which in this case, they look like they're linked together, don't they? So you've got some sort of individual cells, but you've got some that are linked. And then against this very dark background. So this is a negative stain. All has to do with the chemistry of the molecule and the chemistry of the cell wall, outer cell envelope of the cell, bacterial cell. Okay, so to summarize, positive stains color the cells, background clear. Negative stains, cell clear, background colored. And in lab, we're going to be utilizing a variety of basic dyes to perform positive stains. In fact, it's the first stain that we're going to do in lab coming up. And I actually will be using, I believe we use crystal violet, but there's a variety of basic dyes that one could use to do this. But they're going to color the cells. Again, crystal violet's kind of a purple, methylene blue is blue, safranin is red, and malachite green is green. These two types of stains or dyes are used for negative stains, and we'll be using nigrosin coming up in lab. That's going to give us this jet black background. India ink would do the same thing, but we're going to be using nigrosin in lab. These are examples, the positive and negative stains, as simple stains, employing just a single dye, okay? An acidic dye or a basic dye. So they're easy, they're simple. Well, but we're also going to be utilizing different kinds of stains that employ more than one dye. These are called differential stains because they're going to allow us to differentiate or distinguish different aspects of the anatomy, if you will, or cell structures of different kinds of bacteria. Most of these differential cells or stains that we're going to be doing will employ what's called a primary stain and a counter stain. The primary stain is added first, the counter stain is added later in the process. So you've probably all heard of gram-positive and gram-negative, right? Which we're going to talk more about in just a few moments. But this is an example of a stain that allows us to differentiate the purple gram-positive cells from the pink or red gram-negative cells. Acid-fast stains allow us to differentiate between what are called acid-fast bacteria and non-acid-fast bacteria. So we're going to look at two different colors. Endospore stains are going to allow us to see the green endospores, while the vegetative cells that we talked about in chapter 1, they're going to be pink. So we can differentiate endospores from vegetative cells doing an endospore stain. Both, again, all of these actually employ two stains, the primary first stain added, and then a secondary counter stain. And then there are other stains that are designed not to so much differentiate one cell type, say gram-positive from gram-negative, or simply to see the overall shape of the cell, but rather to look at internal or maybe even external structures that certain bacteria might have. And I use the example here of a capsule. You'll be doing a capsule stain, which allows us to see whether this structure is present or not on this outer surface of the cell. Some bacteria are flagellated, some are not. We would perform a structural stain if we wanted to see what sort of flagella are present on a cell. So this slide sort of compares and contrasts the various categories that we just spent the last 5 or 10 minutes talking about. So you can come back to this. We'll be doing these stains, almost all of them, except for maybe these two we probably won't do. But we'll be doing all these other guys in lab coming up soon. Well, let's talk about the gram stain or grand stain, because it is used today, even though it was developed 140 years ago by Hans Christian Graham, another microbiologist. It is a really important stain that all microbiology students perform. It is certainly one that you're going to be doing on your unknown coming up in another couple of months. And it's going to go a long way to sort of begin to narrow down the identification of your unknown bacteria into what's called the gram-positive group or the gram-negative group. And generally, as it mentions here in the fourth bullet, a critical step in identification of many pathogenic bacteria. Yeah, so this is a stain that's performed all the time in diagnostic labs to help us try to figure out what kind of pathogen we might be dealing with. Generally, the gram-negative bacteria tend to be more pathogenic than gram-positive. So let me kind of walk you through the steps, knowing that you're going to hear about this coming up in lab in another couple of weeks, but I think it's kind of important to go through some of these basic steps so you understand sort of how a primary stain works and how a counter stain works and how the gram stain can act as a differential stain, allowing us to differentiate gram-positive from gram-negative. So the first thing you have to understand about gram-positive and gram-negative cell walls is the fact that gram-positive cell walls are thicker than gram-negative cell walls. Okay, so in this right-hand column, you see the thick gram-positive cell wall illustrated by this block, if you will, with the hashed marks. In a gram-negative cell, we have actually several thinner layers of the cell wall. And we've added, after having air dried and heat fixed our slide, the first thing we add is called crystal violet, our primary stain. Now, when you stain either gram-positive or gram-negative cells with crystal violet, you'll notice if you were to examine them under the microscope at that time, that the cells would be purple. And we're going to just depict two rod-shaped bacillus cells here. So you can't differentiate between the two at this point, can you? They're both purple. In other words, the crystal violet gets caught up in the gram-positive and the gram-negative cell walls, as illustrated by these little purple dots. Gets trapped in what's called the peptidoglycan layer of these walls. We'll be talking about this more in an upcoming chapter. After the crystal violet is added, the primary stain, then we use what's referred to as a mordant called Graham's iodine. And what this basically does is the Graham's iodine helps to form what are called complexes within the cell walls of these two categories. Helps the crystal violet sort of stick into the peptidoglycan. So again, if you look and compare the positive from the negative, you can see that the purple's still there. If you examine them under the microscope at that particular point in time, they both would be purple. You would not be able to differentiate the two yet. Now here comes an important part of the stain where we utilize what's called a decolorizer. It's called ethyl alcohol. And we put a little bit of this on the slide. We'll let it run down the slide. And look what happens. The gram-positive cell walls retain the crystal violet, while the gram-negative cell walls release the crystal violet. So there isn't as much peptidoglycan to begin with, and what little did complex with the crystal violet in step 2 is now washed out when the decolorizer is added. So it does exactly what it says it does. It decolorizes, it removes the color from the gram-negative cell walls. If you were to examine the cells at this time under the microscope, the gram-positive would still retain the purple color, while the gram-negative cell wall would be colorless. You wouldn't see any color. It's been washed out. And in the last step of the stain, where we add the counter stain called safranin to both cells, you'll note that the safranin has no effect at all on the gram-positive cell wall because it's already complexed tightly with the crystal violet. While the gram-negative cell wall, which had its crystal violet washed out, can now retain that safranin in a part of its thin cell wall. Okay, So when you examine those cells under the microscope at the end, you can definitely see the purple gram-positive and differentiate it from the red or pink gram-negative types of bacteria. So you simply look at the color. Is it purple? It's gram-positive. If it's pink or red, which is the color of the safranin, it's gram-negative. So some of these stains do employ several steps and procedures, and you're going to be learning about those. These next few slides just go over a couple other stains that we're going to be performing in lab coming up soon. The acid-fast stain is an important stain that allows us to differentiate between acid-fast bacteria and non-acid-fast bacteria. And as it mentions here, acid-fast is going to look pink, the non-acid-fast will look blue. Another very interesting aspect of the procedure of this stain is the implementation of steam or heat to help drive in the primary stain, because usually the cell wall of these acid-fast bacteria is very waxy. And so to help push the primary stain in, we usually use a little steam bath, and we steam our heat fix slides over the steam for several minutes to help drive that primary stain in. So more on that coming up later. But it was originally invented, if you will, the acid-fast stain, to allow scientists to see Mycobacterium tuberculosis, which is the causative agent of TB. It's since been used also to diagnose or identify leprosy. Maybe you've heard of Hansen's disease, it used to be called leprosy. Mycobacterium leprae, which causes this. Mycobacterium tuberculosis causes tuberculosis. And then this other genus called Nocardia. These are the primary pathogens that are identified by utilization of what's called the acid-fast stain. And there's a link there you can look at if you want. You've heard about the endospore. Back in chapter 1, we described it. I had you look at a short little video that talked about how the endospore forms inside a vegetative cell. If that vegetative cell runs out of food or begins to encounter a harsh environment, remember, the endospore is there to sort of ride out the bad conditions until conditions improve. Then it germinates back into the vegetative state. So the production of endospores is a really interesting and important mechanism that some bacteria employ, not all, but many. Some of the bacillus genus, in fact, tend to be endospore formers. This, like the preceding acid-fast stain I described, is going to employ steam to drive the primary stain into the endospores, because their structure is different than the structure found in the vegetative type of cell or form of the cell. And so this stain allows us to see the endospores. They're going to look green, while the vegetative cells are going to look red. And so, as I mentioned here in the next bullet, there are a variety of different kinds of bacillus bacteria that we're going to be utilizing in lab, some of which are pathogenic, like this guy that causes anthrax, and also Clostridium. Maybe you've heard of Clostridium. Clostridium is a very nasty anaerobic bacterium that can cause tetanus and gas gangrene and botulism, very, very nasty things. It can ride out harsh conditions by forming endospores. But we're going to be using bacillus in lab and looking for endospore formation in bacillus. And here's another link again that talks a little bit about that endospore stain. So more on that coming up in lab in several weeks. Okay, I'm going to stop here. We'll call this part 1 of 2 here for the chapter 3 material. I'll follow up with part 2 of 2 after this.